Welcome to Breakthroughs from Science Mission. I'm Sadashiva Pai and going to discuss a couple of breakthrough papers published in the week of August 25th. Let's start. First, we'll discuss a paper published by Gemina and colleagues in Cell that demonstrate retrograde neurodegenerative signal transduction from axonally synthesized transcription factor ATF4. Why is this paper so important? The role of intra-axonal protein synthesis in neurodegenerative disorders remain unexamined. Authors show that axonal translation is activated in response to A-beta 42. Axonal ATF4 synthesis is required for the retrograde spread of A-beta 42 induced neurodegeneration. Axons in the brain of AD patients show more frequent localization of ATF4 protein and mRNA. Let's look at the data. Authors used microfluidic chamber to separate cell body and axons to evaluate effect of amyloid beta peptide. Applying A-beta 42 to axons and cell body separately resulted in non-significant increase in translational markers, protein EBP, and significant increase of SP6 in axons, while no change in EBP and decrease in SP6 was found in the cell body. Scrambled peptide and A-beta 40 had no effect. Protein synthesis inhibitors prevented the change. 48 hours of A-beta 42 exposure, the number of tunnel positive neurons was significantly greater, whereas axonal fragmentation was not induced. And immunostaining of A-beta 42 was only seen in axons and not in soma, indicating origination of effect from axons. Application of protein synthesis inhibitor anisomycin to the axon significantly decreased A-beta 42 induced axonal tunnel staining when applied early, while retrograde transport inhibitor ciliobrevin inhibited the protein transport when applied late. RNA sequencing analysis revealed high levels of ATF4 expression in axons, but not in soma after A-beta 42 application to the axons. ATF4 protein levels reached peak after 6 hours of application and were back to control levels after 24 hours. Protein synthesis inhibitors and ATF4 siRNA blocked increased in ATF4 in the axons. ATF4 transcriptional target protein CHOP was increased in cell bodies 48 hours after treatment of A-beta 42 to axons. This increase was blocked by retrograde transport inhibitor and ATF siRNA applied to the axons. Also, application of ATF4 and CHOP siRNA to axons prevented cell death induced by CHOP. Next, authors induced semi-acute amyloidopathy in mice by injecting A-beta 42 oligomers into mouse dentate gyrus and were able to demonstrate increase in ATF4 mRNA 2 to 7 days after injection. ATF4 protein levels were increased and was blocked by siRNA. CHOP levels were increased after 7 days, and authors also demonstrate cell death in vitro hippocampal cultures. Further, authors were able to also show the increase in ATF mRNA and protein levels in human brain samples of AD. Thus, authors were able to show A-beta 42 could increase transcriptional factor levels in the axons, which is transported retrogradely into the cell bodies and kill the cells. Next, let's look into the paper published by Nicola and colleagues in Cell. Authors demonstrate circulating tumor cell clusters are more potent in forming tumor metastasis. Why is this paper so important? Circulating tumor cell clusters are present in blood of patients with cancer, but their contribution to metastasis is not well understood. Authors show that CTCs are highly metastatic compared to single circulating breast cancer cells. Authors identify plakoglobin as the key mediator of tumor cell clustering. Knockdown of plakoglobin expression in mouse model suppresses CTC cluster formation and reduces metastatic spread. Let's look at the data. Authors injected 1:1 ratio of lung cancer LM2 cells labeled with GFP and M-cherry into the mouse mammary glands and found that 2.6% of CTCs in blood were clusters and 97.4% were single cells. Among the clusters, 91% were dual positives for both M-cherry and GFP, indicating their origin from neighboring cells within the primary tumor. Analysis of metastatic deposits revealed about 50-50 distribution of single to both labels, and further calculation indicated that CTC cluster is about 50 times more likely to give rise to a metastatic deposit than a single CTC. Authors injected single and cluster CTC into the mice separately and found that not many single cells survived intravasation. They also found increased in caspase 3 activity in single cells, and these mice survived longer than cluster CTCs. RNA analysis revealed and confirmed in breast cancer patient of high expression of plakoglobin in tumor cells. shRNA mediated plakoglobin suppression triggered disruption of cell-to-cell contacts in 6 out of 7 breast cancer cell lines grown as a monolayer. Plakoglobin knockdown in 2 tumor cell types dissociated clusters into single cells. Inoculation of cluster and single cell preparations were comparable in producing a reduced number of lung metastases. Plakoglobin knockdown did not alter primary tumor growth rate, nor did it affect the total number of single CTCs derived from the primary tumor. In addition, bioluminescence imaging of mouse lungs demonstrated a striking 80% reduction in lung nodules for mice bearing plakoglobin suppressed primary tumors. WB analysis from patients revealed presence of various amounts of single cell and cluster CTCs in the sample at different time points. Patients with cluster CTCs across more than 3 time points had a mean progression free survival time of 32 days compared to 135 days for patients where CTC clusters were found during 1 to 3 time points, and 161 days for patients with single CTCs only. RNA analysis revealed a very high expression of plakoglobin in CTC clusters than single cells, with significantly reduced distance metastasis free survival in patients. Authors have a model schematic showing that high plakoglobin regions in primary tumors are likely to generate CTC clusters with increased metastatic potential. Thank you for watching Science Breakthroughs from Science Mission.